about doing research, going to grad school, check it out. Next few slides. Maybe I turn on the right thing. See here. Or not. All right. Small technical difficulties. One moment. Alright, well, we're doing my remote's not working, so we'll make it work today. Alright, so a few items of review. I know that some people were asking their TIs this weekend, and I just want to make sure that everyone is clear. Remember, you need to know the 3 types of microtubules and the 3 types of motors associated with those microtubules and how they're moving. In particular, there was a question about what was on the kinetic core. So remember, we talked a little bit about this, but sometimes there's a motor there and sometimes there's not. Well, that doesn't have to be one of the 3 that you know. I'm mostly concerned with what's happening at the kinetic core. What motor is involved in moving the kinetic core?
Modified kinesin and dynein.
So modified kinesin and dynein, which one is doing the moving? Dynein. Which one is doing the anchoring? The other one, the modified kinesin. Right? Dynein is pulling that towards the negative end. Kinesin is just hanging on. Okay? All right? Don't forget that you have a review session tomorrow, 5:30 to 6:30, this room. All right, your TIs will be here, and it is your chance to ask questions, clarification, go over material. Come prepared to ask them things. Otherwise, you guys will just be standing in a room together. Okay? So you have a chance every week to work with them in recitation. The review session is for you to get any last-minute answers, questions answered. More than just, is this on the exam? Because they don't know. Only I know that. So ask them questions about content that you're unclear about, like the kinetic core motor. Okay? All right, so today we are going to finish up, well, get as far as we can with DNA replication. Whatever we get through by the end of today, that's where the exam material cuts off. All right? So for your homework, sorry about the traffic jam. I lost track of the table. Make sure you get that in before you leave. Was to help you look at the material before today, so you're at least familiar with it. You have a little more time working with that before the exam. Right? So we're going to get through replication. You need to know about replication forks and the enzymes involved in DNA replication in the cell. We talked about it in a test tube last week. One other point of clarification, just to make sure that we're on the same page. Talked about the direction of that different things are moving in the process of replication. Synthesis versus polymerization. So what is polymerization specifically? Can anyone define it? The addition of the nucleotide bases to the synthesized strand. Specifically, what happens between those 2 bases? phosphodiester bond is formed by a condensation reaction. So it's that condensation reaction that's happening from the free 3' OH to the incoming 5' phosphate. All right, so polymerization refers specifically to that reaction. Synthesis refers to building the strand. All right, so I just want to make sure everyone's clear between those two terms, synthesis versus polymerization. All right, so this is where we ended last week. We were talking about PCR, polymerase chain reaction, the minimum required things needed and how that worked. We know that we need a primer, a template, a polymerase, and some nucleotides at a minimum. We stick a primer down on our template. The polymerase comes in to that free 3' hydroxyl group and begins building a new strand complementary to the template. All right, so our end product looks something like this. Remember our notation for the DNA strands and their orientation. All right, so for the exam, I should be able to give you any combination of template and primer, and you should be able to draw me the schematic of what the final product is. All right, so let's take a look at what I mean. So if I give you this combination of primer and template, can you draw the resulting product, double-stranded product? Take 10 seconds and do it. All right? We'll go through a few of these just so you get the sense of what it is that I mean. All right, I've given you primer, template, polymerase, and the nucleotides. What does it look like when the polymerase is done? Take a look. All right, everyone had a chance to sketch that out. It should be pretty quick. Polymerase sits down on that free 3' end, uses the template, copies. That's what the end product looks like. All right, everyone see that? There's no free 3' hydroxyl on that other end. It can't copy the template. All right, let's do just a few more examples. What about this? Take 5 seconds and sketch that. What's the final product? Right? That free 3' hydroxyl copies the template all the way down. You get a complete double-stranded piece of DNA after you're done. What about that one? What's the final product look like? That's you. Remember how polymerase acts. All right, everyone had a chance to sketch that one? What does it look like? The same, nothing changes. There's a free 3' hydroxyl there. There's no template, it can't do anything. Free 3' hydroxyl there, no template, can't do anything. Right? It needs both that free 3' hydroxyl and the template. So it looks exactly the same after polymerase is acted. What about that one? What does the final product look like? All right, let's take a look. Free 3' hydroxyl and template, it can extend. Free 3' hydroxyl and template, it can extend. All right. So you should be able to do any combination of primer and template that I give you and draw me what the final product is, whether that's a complete double-stranded piece of DNA, no change, or some partial copy of that template. All right? Everyone clear on that? All right. So that finishes up DNA replication in a test tube where we're copying small stretches of DNA. Let's take a look at how this translates to DNA replication in a cell, where we're copying millions of bases rather than just thousands. All right, specifically, we're going to look at how double-stranded DNA is replicated in the cell, and we're going to look at the system in bacterial cells to start off with. So, there are some YouTube videos. I'm not going to show any of them today. Go ahead and take a look at them. There's some great molecular animations of the process of DNA replication. I'll post those on Facebook, and they'll be in the lecture slides that I post today. But let's think about quickly, we're talking about copying millions of bases. Bacterial cells can divide fairly quickly. We don't want to take 5 years to replicate every cell. So the amount of information we have to duplicate, how fast are they doing this replication process? How fast is DNA being synthesized? To do a quick calculation, we have a cell with 1 million base pairs. They divide every 20 minutes. What is the rate of DNA synthesis? All right, how fast does DNA replication happen? Everyone had a chance to do that calculation? All right, let me see if the clicker base is working at all today. All right, so how fast is that happening? Not working? All right. You guys get a freebie day then. Oh, wait. Get it. Really slowly.
This is my base.
The mobile is working and not the iClickers. All right. Well then, everybody gets a freebie day. I'll figure out what's going on for Friday. My iClicker, Monday morning, my base is not woken up yet. So, all right. So how fast is that? 833 bases. So almost 1,000 bases per second in order for that cell to replicate every 20 minutes. All right. So replication has to happen quickly. Think about larger genomes, size of a human, how quickly things have to be replicated. Think about how important your DNA, that replication process has to happen accurately. All right, so this is a pretty astonishing molecular machine that's doing this replication process. Let me get rid of this. Lots of technical difficulties. Okay, there we go. All right, so again, we're going to look at that DNA polymerase. So how is the chromosome replicated? Where does replication begin? How is that polymerase acting in the process of replication? And where does replication end? Probably get to that after the first exam. All right, so we're just going to get to where does replication begin and how does it proceed. So you have to know, you had to look up for your homework, the replication enzymes. So there are multiple polymerases involved in replication in the cell. We're going to look at 2 in bacteria. There are bunches more in eukaryotic cells. Specifically, we're going to talk about today polymerase 1 and 3 and their roles. Helicase, single-stranded DNA binding proteins, DNA gyrase, which is a topoisomerase, and DNA ligase. All All right, so there are lots of components to this molecular machine that's doing the replicating. All right, what else do we need for replication? Mumble, mumble, mumble. Nucleotides, right? We need the building blocks. What else do we need? We're thinking about the test tube. The primer. All right, in the cell, in a tube, we're adding to everything in there for a specific reaction. In the cell, we need something to make that primer for us. An enzyme called primase. All right, and unlike the test tube where we're using DNA primers, in the cell it's an RNA primer. All right, that's what it looks like in the test tube. DNA, we're replacing that in the cell with an RNA primer provided to us by an enzyme called primase. All right, it serves the exact same function. It provides a free 3' hydroxyl group for polymerase. All right, so those are the basic enzymes we need for replication in the cell. If we look at the process again using our schematic, we have a double-stranded piece of DNA. We start from one end, say it was a linear chromosome. Gives us 2 templates to work with. We have to put down that RNA primer with primase. And then the polymerase can come in and replicate, synthesize based on that template. So that's one strand. Notice the directionality that things are happening. All right, replication is all about keeping track of what direction you are synthesizing your DNA in. So on the other strand, same process. Put down our primer with primase. Polymerase can come in and replicate, synthesize that new strand of DNA based on the template. If we want to copy this whole chromosome, we open it up a little bit more. On one side, the polymerase can just keep on going. Right? It's opening in the direction that DNA is being synthesized. That strand is called the leading strand. The template it's being based on is the leading template, or leading strand template. All right, this strand, the polymerase can sit down on that primer, and it can just keep on going. So it is synthesizing in the direction of that wishbone shape called the replication fork. So this is the replication fork. The leading strand, the polymerase is synthesizing towards that replication fork. So as new DNA template is opened up, as we continue to pull apart that double strand, it can just follow along behind and synthesize a new piece of DNA. On the bottom strand, it's a different situation. It's being synthesized away from the replication fork, which means that as new template is opened up, the polymerase can't, there's no free 3' hydroxyl for the polymerase to sit down on and begin synthesis. All right, so this strand that is being synthesized away from that replication fork is called the lagging strand. And the template it's being built off is the lagging strand's template. Lagging because every time a new stretch of DNA is opened up, we need that primase to come back, put down a new primer, so that polymerase can continue to copy the template. All right, so this, we can call this the half bubble. We're looking at half of a replication bubble. You should be able to identify, given any sort of representation that I give you, what is the leading strand and the leading strand template? What is the lagging strand and the lagging strand template? So let's look at a few examples just so you get an idea of what I mean. All right? So since we don't have clickers, we'll have to yell out the answer. So which one of these is the leading strand template? C. Excellent. About here? B. B is the leading strand template. What about here? All right, how do you know? Are you sure about that?
B.
All right, hands for A, hands for B. Why B? Right? The leading strand is synthesized in the direction going towards the replication fork. What direction is it synthesized in? 5' to 3'. What is the orientation of the template? 3' to 5'. Right? So the primer, if you put a primer on the end of that, polymerase is going to move towards that replication fork. What about this one? B. So all of these representations, right, particularly that one I just showed you, you have to keep track of orientation in order to be able to figure out which the leading strand is. What direction is the leading strand synthesized in? Hands for A. Hands for B. Hands for C or D. All right, it's 5' to 3'. That's the leading strand. What direction is the lagging strand synthesized? Shout it out. 5' to 3'. DNA is always synthesized 5' to 3'. That's why we have the lagging strand problem. Right? Remember our DNA structure. The strands are anti-parallel. As you pull them apart, polymerase only goes in one direction, so it copies the strands in opposite directions. Okay? So let's look at the enzymes and the roles that they play. So I gave you the list. What does each one of those do? So in addition to the polymerase, which we know is polymerase, which polymerase is it for bacteria that's copying the template? Polymerase 3 in bacteria starts at that primer and copies the template. In addition to that, polymerase 3, we have the helicase enzyme, which sits right at that replication fork and opens up that double-stranded DNA structure so that you have a single-stranded template for the polymerase to work on. Right? It requires energy, it's driven by ATP, and it just unwinds that DNA. And we know double-stranded DNA wants to be in a double helix conformation, so if we pull it apart and there's a complementary base pair nearby, it's going to want to form those hydrogen bonds again. So in order to hold the double helix apart, for the polymerase to work on it as a template, we need to stabilize it. So those single-stranded DNA binding proteins stabilize the DNA so that it doesn't just reform a double helix after the helicase has unwound it. Right? So the polymerase has time to sit down on that single strand and copy it. We also have a topoisomerase. In bacteria, we call this gyrase. It allows the DNA to swivel. So you can imagine you have a double helix, two strands coiled around each other. As helicase is pulling that apart, you are condensing all of the rest of that tension into a smaller and smaller piece of DNA, right? And to the point where it's so tight you're not going to be able to unwind it anymore unless you relieve some of that tension. So the topoisomerase relieves the tension created by the helicase pulling apart that double strand. So those are the enzymes we've looked at so far. We have the piece of cheese helicase that's doing the unwinding. We have the grapes. I must have been hungry this morning. All right, it looks like they all look like food to me. So the grapes are the single-stranded binding protein. And then that half-eaten donut, which some of us might have had for breakfast, that's our topoisomerase, and that's relieving the tension created by the helicase pulling apart the DNA strands. Okay, so we're still missing a few things to complete replication on both strands. So first of all, if we think about a chromosome and how long it is, does replication start at one end and proceed all the way to the other, linear fashion? No, it would take too long. We have too much DNA. All right, so rather than this half bubble where you start at the end and pull it apart, what actually happens in cells are full replication bubbles. Right, so the helicase starts in the middle of the chromosome, creates a bubble. And because it's pulling apart in the middle of the chromosome, replication can happen in both directions. So if we stick our two, basically replication bubbles, two half bubbles stuck together, right? So if we look at that bubble, it has an inverse symmetry. On both sides of that bubble, you have a leading strand and a leading strand template. You have lagging strands and lagging strand template. And you have a replication fork on either side. So any place that the DNA gets opened up, you're going to have machinery moving away from that point and replicating the DNA. Excuse me. All right, so we looked a little bit at the half bubble model. Similarly, for any full bubble, full replication bubble, you ought to be able to identify where the leading strands are, where the lagging strands are, where the template for each is. All right, what is that arrow pointing to? Leading strand. That one? Lagging strand. That one? Lagging strand template. Leading strand template. Pretty straightforward, right? So if I give you, or just ask you to draw a replication bubble, you should be able to do that. Right? Putting in the leading and lagging strands in the proper orientation. Right? Remember to keep track of your orientation. Students often get mixed up in exams when they're nervous about which way things are going. Remember to keep track of what way things are synthesized and what way the replication bubble is oriented. Okay? Any questions so far? Everyone have replication bubbles down pat? No problems? No one's going to miss that question on the exam? All right. I'm going to hold you to that. All right, so there's one last piece of replication to talk about before we're done with material, new material for the exam. All right, and that's Okazaki fragment. So specifically, let's go back to our half bubble. When we're looking at the leading and the lagging strands, the leading strand just can keep copying as more template is opened up. Just keeps on going. that lagging strand is creating short little fragments of DNA. Those short fragments are Okazaki fragments. And there are many things named after the person who described them. All right, so we have a problem on the lagging strand in that what we really want is a continuous copy of DNA. What we have is short fragments with bits of RNA in the middle and a gap. All right, so how do we get these Okazaki fragments put together so that that lagging strand is also a complete copy?
Polymerase 1.
Polymerase 1 and ligase. All right, so we have those triphosphates on the end. What we want is that DNA to replace and displace that RNA primer. And then, if we can do that with polymerase 1, we have DNA, but we still have that nick, that break in the single strand, the backbone of that single strand, in the phosphate sugar backbone. All right, we have all our bases present. We have a 5' phosphate, we have a 3' hydroxyl. We just have a break. So we have DNA ligase with the help of ATP can seal up that break. All right, so we have our DNA polymerase 1. Polymerase 3 does the copying. Polymerase 1 for the lagging strand comes in, removes that primer, fills the gap with DNA, and then ligase seals the nick. So on the lagging strand, we have that extra step with polymerase 1 and ligase needed to complete replication of a complete strand. All right, are there any questions about DNA replication or about any material covered for the exam? DNA polymerase 3 in bacteria is the one that sits down on the 3' hydroxyl of the RNA primer and does the synthesis for both the leading strand and then the fragments of the lagging strand. Polymerase 1 comes in and replaces the RNA primers on the lagging strand. Eukaryotes have multiple polymerases as well, just to be difficult, they're called different things. We'll talk about those probably after the first exam. Are there any other questions about replication? Or anything else on the exam? Now's your chance. Yeah. Yes. So the question is about the full bubble of replication and whether both sides have Okazaki fragments. Go back to that slide. All right, so both sides have a leading strand being copied, and both sides have those Okazaki fragments on the lagging strand. Right? It's because the process is the same, right? So on both sides, you have a replication fork. It's just inverted in orientation, right? So remember that the way DNA is synthesized is 5' to 3'. So you're always going to have a strand that's being copied away from that replication fork. Yeah. Organelles. What do you need to know for organelles? So you need to know organelles and their function and be able to rank them by size like we did in class. Right? And where I have specifically in homework or class stressed the size of something, like double helix, or one turn of the double helix, know those sizes. All right, so.
I got a question.
Question in the back? Yeah.
Could you go over the hydrogen protons or the hydrogen donors and acceptors?
Hydrogen donors and acceptors. How do you tell if somebody's a hydrogen donor or a hydrogen acceptor? That's the simplest way. If it doesn't have hydrogen attached to it, it's probably an acceptor. If it does have hydrogen attached to it, then it's probably a donor. Right? It's based on the electronegativity of those atoms as well, but that's the simplest way. All right, so when you're looking at functional groups, do they have a hydrogen to give? Any other questions related to the exam? All right, so on Friday, I know you don't want to think about after the exam right now, but we're going to pick up with what happens at the end of replication. All right? Don't worry about that now. We'll talk about it on Friday. One more question?